first application session and directly opening the second one. So we will have again a set of 3 talks. Sorry. So now the second session, the topic will be on SARS-CoV-2 protein sequence diversity by Li-Chun Chong.
Thank you for the introduction. Hi everyone, I'm Li-Chun Chong from Malaysia. I'm currently a PhD candidate in Germany. So this is a project, extended project from my master thesis. This work is about the application of our published alignment-free 2 unique means that on the SARS coronavirus 2 dataset. This was achieved through a process akin to data compression. As we all know that the data tsunami, so thanks to the advance in the genomics and proteomics approaches, the concerted sequencing efforts of SARS coronavirus 2 have remarkably reached a historical milestone in viral sequencing as the fastest and the single largest source of the data growth. So for the proteins data that are first 8 months in GSAID and the first 20 months in NCBI viruses has exceeded the total number of the protein sequence of all the viruses since 1977 to December 2019 before the COVID-19 pandemics. So this is one of the plots that from our manuscript that currently under review. So traditionally, we know that the alignment-dependent approach is largely used to analyze the viral sequence diversity. However, the increase of the SARS coronavirus 2 sequence have limited the alignment-dependent approach, and because it is generally time-consuming and the compute really need a very high compute resources. So that's why we need an alternative alignment-free approach. So towards this, we try to develop an alignment-free 2 unique means, and it is accessible through this QR code. Unique mean perform an exhaustive search through the data, the protein sequences database, and then try to generate the minimal set. So let's look into the concept of the minimal set. This concept actually is the key to the unique mean ability to compress the biological sequences. So let's say we have these 3 unique sequences in our initial data set. We then try to generate all the overlapping k-mers from each sequences. And then in this example, we are trying to use 9-mer for the immunological applications. From here, you can see that although 3 sequences are each unique, but they share some of the some identical 9-mers that that in the light green color and the light blue colors. So from here, we can see that we can say that the first sequence actually is redundant, and the second sequence and the third sequence will be represent the minimal set of the initial data set of 3 sequences. So in this example, we try to compress the data set by removing one of the sequences, but in large data set, actually the compression can be higher. So now we already covered the core idea of the unique mean. Let's look into how the idea implement into unique mean. From this figure, we can see that the unique mean comprised of 5 steps. The first step is the generation of the overlapping k-mers. Second step is the frequency grouping of the generated overlapping k-mers. The third one is identification of the pre-selected minimal set of sequences omission of the cameras connected to the pre-qualified minimal set sequences, and lastly, identify the minimal set of sequences. This approach has been applied as the pilot study into the non-redundant dataset of dengue as of March 2018, and also all the viruses as of September 2018. So a significant compression was achieved for each dataset, which is about 44% for the non-redundant dengue dataset and 38.5% for the all viruses dataset. For a detailed protocol how to use unique mean, you can check out our recently published bioarchive. So we have covered all the basics. Let's move to the highlight of this talk is the application of unique mean in SARS coronavirus 2 dataset. So all the protein sequences of SARS-CoV-2 were retrieved from the GSAID EPICOV as of July 2021. The compression was achieved by subjecting the data sets into two levels of the data compression. The first one will be the remove the redundant sequences. So it's considered a deduplications, which we expect in the public databases. And then we try to remove the non-redundant sequences using unique mean to generate the minimal set. So for this, we try to apply to all the proteome and separately for each sequences. About 56 million of the protein sequences were retrieved from GSAID and deduplicates and result into about 1 million of the non-redundant sequences. After that, we try to further compress using the unique mean and result in 273,851 protein sequences in the minimal set. So this actually is just about 0.5% from the retrieved data set. In summary, so we can see that is only less than 0.5% of the reported SARS-CoV-2 protein sequences are required to represent the inherent viral peptidome diversity. But when we try to apply to the higher taxonomy, which is the family rank, we actually achieve a mere increase about 2%. So this finding actually is important for the implications in design of vaccine, drug, and diagnostic, whereby the number of sequences required for this study actually will be drastically reduce the discovery process, and then but it's still providing the systematic evaluation and the coverage of the pathogens diversity. For the future direction, we are planning to apply the comparisons, two comparisons. One is the time point comparisons between the previous work, July 2021, and try to update with the current SARS-CoV-2 data set. And also would like to see what we expect is really for the July 2022 data set. The next one, we would like to compare between the variants being monitored, alpha, beta, gamma, and the delta variants with the VOC Omicron variant. So that's all from me. Thank you very much. Before I end my talk, I would like to acknowledge Liner and Dr. Asif on contributing the unique mean development. And also thanks Robert and EpiBioNet provide the financial assistance for my ECCB trip. Thank you very much.
Thank you. Questions? Okay, so maybe I can start. Can you dig a bit on this comparison of the different variants with your method, what you could help finding with that?
Oh, like Because we know that maybe difference of the variant have their own diversity, and then from there we can see how many, what's the pattern of the compression, and then yeah, from there we can see the diversity as well, and then also we can know that actually maybe we no need to use all the sequences for the vaccine discovery or maybe drug discovery, because if we want to plan to search for the conserved region of the, for example, for the vaccine discovery, we really need to look into conserved region. So you actually no need to go through all the sequences in the database, but you can just directly grab some of them, yeah, based on the unique mean.
Yeah, that would be directly draggable or something.
Yeah, yeah. It will reduce the time. As you know that now a lot of the tools is not big data ready. Yeah, so.
And did you try to correlate the protein data with the sequence data, or the sequencing data, the RNA-seq data, or
No.
No, you didn't try to correlate.
But actually, from what we expect, actually it will be almost the same, but we not done on that way yet. Yeah.
Okay, thank you. Yeah, please, thank you.
So the SARS-CoV-2 viruses, they don't have a lot of mutations against each other. Do you think your method would also be applicable to, like, for example, HCV, where we see a lot of differences between different viruses?
Yeah, we actually have another paper that we are preparing that we already tried to apply for all the virus families that known. But the data actually is before COVID-19, so we are planning to update with the current data, because my master actually is started in 2018 or 2019, so that time we are trying to start with that. But maybe we need to really update with because of COVID-19. Yeah, yeah, we have done that. Maybe you can follow the unique mean, and then we will update that.
Okay,
yeah.
Thank you.
Other questions? Okay, so if not, we will thank you again.
Thank you very much.
And our next speaker is on patient cancer stratification, and it's Jake Chen.
Right. So is this the slide to advance the slides? Okay. Okay. All right, good morning, everyone. My name is Jake Chen from the University of Alabama at Birmingham. My student couldn't be here, but this is the work over the summer 2 days, plus some discussions in something that I organized called the Data Science Hackathon. And I think what we're really trying to do in the lab overall is to take the multiple omics data and connect it to precision medicine. And then there are multiple steps. Of course, you can do it in a way that is only using machine learning, knowledge agnostic way, but we tend to favor layering this predictive model with information curated from biological literature or high throughput data, network data, and build simulations. And this way, these whatever we build can be interpretable and validable. And that's the overall mission of the lab. And for this particular study, I'm using a data set relating to glioblastoma multiforme. We have collaborators that's in the cancer center. They're doing a lot of radiation treatment of the brain tumor. This is the most aggressive form of brain tumor. Average survival time is 12 to 15 months. Very difficult to treat. And because it's in the brain, many of the drug cannot get into the brain, so you have to have very limited option to sensitize the tumor and use radiation. And so a lot of research are needed to really find out what form of the tumor are more aggressive, and then you treat it more aggressively. Right, so there's a lot of work that's been done on the aggressive high-grade gliomas in general. Microarray-based gene signature approach has been around for many years, almost 16 years. So the approach is usually you sort the samples and then genes, you find, use a clustering to find gene signatures. And these gene signatures, for example, the proneural subtype gene signatures, are associated with better survival. And then some of the mesenchymal proliferation type may have more worse survival. So this is what gene signatures are used for. And why can't we just directly use this approach? And one of the inspirations is that a lot of the time we know that the samples are very heterogeneous. And then if you just treat the sample as if it's just a homogeneous set, you may not get the good result. For example, there's a paper published on not in cancer called the COPD, it's a lung disease. And if you look at the embedding and look at the clinical subtype called never smoker, few smoker, and they're all over the places. You really cannot sort them out in the embedding. But I think they did try a different clinical information. We call it clinotype. And then you can see that roughly the sample can be grouped. Right? When the sample can be grouped, and what they're doing is to really put them into 3 clusters, and each of the cluster may be enriched with a gender clinical subtype. And then they do the clustering. And when they do the clustering, you have C1, C2, and C3 cluster. And they're actually a C3 cluster. You have mostly the male, and the C2 cluster are mostly male. And then you have this essentially clusters that are gender specific, and then the signatures are quite different. So if you really want to find precision medicine tailored treatment, you really need to first find the precision biomarkers. And these biomarkers can also be heterogeneous. You have to use clinical information, which we call clinotype. Right, so we started with that question. We say, well, how do we initially come up with a very good knowledge base that capture all of the GBM information? So we have to go to TCGA and get the sample subtype information. We get the gene mutation information from COSMID database. TCGA, we have to process them into differentially expressed genes, and we will go to drug bank, and also we use disease survival, DSS, disease-specific survival information. In the end, we come up with 900 genes, and then we embed them. And when we embed them, we actually use a software that we built called SEAS, Statistical Enrichment of Analysis of Samples. What the SEAS can do is not only to show the embedding, but also allow you to explore all of the available clinical information in a figure such that you can label, say this is cohort, this is a population background. If you select a cohort to be, this case is green, and you say, well, this is a specific subtype, you can really see how it's distributed and what does the neighborhood look like. And then the software also can automatically find enriched for a particular neighborhood, what are the clinotype that's enriched, and then do survival analysis. So this really help us to explore, but it doesn't change the coordinate. So then what are some of the interesting things that we can do? For example, you can see that these cyan-colored clusters are patient-derived xenograph models sequenced from UAB. When we embed it, you see that they're distributed around the neighborhoods of TCGA samples. Right, so there's one particular one that's called X1516-1, and it's in the neighborhood. And we look at the neighborhood and find what is the enriched disease-specific survival date. right? Because we would assume that the survival date of the neighborhood probably could tell some information about this sample, right? So we look at all the survival date, and then you can average. Mean is 242 days. In fact, if you look at these three, X15, 16, 1, 2, and 3, across these, they're pretty consistent. The embedding is neighborhood is consistent. And when we apply the radiation treatment, they don't move. So that means that this is the previous sample is really an example of radiation-resistant tumor, and it's very difficult to treat. And in this, another example, we find a different PDX samples, and after radiation treatment, the profile changed, and the embedding also changed to a different neighborhood. When we look at the embedding neighborhood of the before and after, we see that there's actually longer survival time. So from this simple analysis, all of a sudden you find a clue that these embedding neighborhoods are important, clinical attributes are important, and then combining everything together, you can do interesting things. So we're not happy with the original embedding because the original embedding, when we find the subtype, and they're all mixed. So we use a smart trick, well, not a simple trick, to essentially measure the in-cluster distance using this IMSD measures. And then we say, well, can we find a weighting scheme so that we can group the embedding clusters around certain subtypes of the disease? In fact, this is the original embedding. You can roughly see, survival and the low. If we do that, you can somewhat differentiate based on this heat map. But then you can't really differentiate the subtype and the survival. Right? So if you use that and choose, this is ISMD is somewhat minimized and refined, choose a weight to do this weighted embedding, you can actually separate the subtype, different colors, and also the low and high survival can also roughly be in different neighborhood. This would really allow us to do the C's type of analysis, right? So with that in mission, so we did actually look at all of these sample enriched gene signatures, and then we did the pathway analysis to see in proneural, mesenchymal, and classical, what are the pathways that they have in common? There are 12 in common. What are the pathways that's very unique to each of the specific subtype? And from here, we were not satisfied, and we decided to use some of the tools that also developed in our lab called the Pager. Pager is a compilation of all the gene set, pathways, and network modules. And one thing that we did uniquely from other, like MCDB, is that we have this co-membership-based pack-to-pack relationship. And then we also have the R-type pack-to-pack relationship. And you can read the papers to just understand how we did it, but essentially, we do have those representation. So we can actually look for the enriched pathways and overlapping with the relationship to build a network that essentially tied all of these previous results together into a so-called pack-to-pack co-membership interaction map. Right? In this interaction map, you can see different regions, and then they're making sense. But all of them are connected. That's one message, even though there were subtype-specific gene signature, but then there are also common ones that are connected. Especially if you look at the color, some of them are shared, some of them are unique. Like classical, or a lot of them are just all over the places. Then we want to ask the question, can we even do better? Infer, maybe have a knowledge graph that's mapped to the cellular substructure. In fact, we use the gene ontology information, and we use the pack-to-pack network that I showed earlier to filter the gene list and build a knowledge graph that looks like this, right? So you no longer can just use machine learning alone, but this really allows us to traverse the path. What is the starting point of the signaling cascade? What is the ending point? Where does it land, right? Some of them lands in the preneural. Does it actually up or down regulate the genes, and so on and so forth. And we want to find the path that is more likely to be the path happening in a specific subtype. And I won't bore you the detail, but essentially we use another tool called BEER that allow us to construct the literature validation of significant co-citations of the knowledge graph, right? And then we can build that list. So this is an example of what you can do in BEER. You just enter the gene list along with some terms, and then you can build a knowledge graph that not only involves genes, but also the directionality of the citation, and each edges is a significant citation. And also there are terms that are actually gene ontology terms, and so on. So with that, and then finally, we can build gene signatures. The gene signature that we built here are all related to the knowledge graph that we mentioned, we mined. And so these are the subtype-specific gene signature that we found. We're thinking about submitting to a journal. So these are still unpublished. And many of the things that we have found here, I don't have time, and we actually can find a mechanistic link of these signatures to the disease. And if you read the literature, many of them cannot be validated. And the performance are pretty decent. The overall ones is beating each of the individual genes if you want to do the survival curve analysis. Okay. I think I will skip this, and this is a further validation how we validated that they're biological relevant for the survival signatures. But I think, yeah, so let me just go through here. The discussion is, I think it's important to build subtype-specific, clinotype, enriched gene signatures if you want to truly have this precision in this type of clinical applications. And interpretability is very important, because if we don't have that, our collaborators would be very hesitant to use it. And they say there are so many of them, why should I use it? And so potentially, where implementation, we cannot validate it directly in the wet lab, but that we can validate it using knowledge graph. With that, we want to thank these collaborators and the funding agency, and you can play with the software if you're interested. Thank you.
Thank you. We have time for questions. So, okay, I will start. So you have many different tools. What I really like is the fact that you try to combine data-driven and some knowledge-driven ideas. So I think this is a very interesting point. I would like to have, how would you do if you work with a disease where you don't have a lot of clinical data, a lot of metadata, like you have to do something unsupervised completely? So what would be your approach or your philosophy for that?
Yeah.
Like not cancer.
Yeah, that's a very good point. I think there are lots of rare diseases, and we don't work in those areas. There are probably other approaches would be important. But essentially, if your AI is smart, you can generate some candidate, and then if you can look for literatures for maybe a gene or disease that have similar phenotype elsewhere, and then bringing those information should be helpful to some extent understanding the problem at hand.
Okay, and based on your experience in the first part of what you were presenting, like you have your embedded data and you try to visualize the different clinotype, what is usually what is separating the samples? Because we saw that smoking, non-smoking was not so much splitting everything, but the genders, yes. And so in your experience, it's usually the same way? Or what type of clinical data are usually the first, or at least on the first two axes of the, of the dimension reduction?
Yeah, I think that's a very good point. Actually, there's no known answer. That's why we bring in all of the clinical attributes from TCGA. That's up to 200. And you can explore them by saying, well, I think that my collaborator said chemotherapy is important. And in that tool, we can not only see the label, we can also see the density distribution. And so you explore, you think, well, maybe I should use this and incorporate this, and then just do enrichment and also go to the neighborhood. This is very exploratory.
Yeah.
Right.
Now, I was also thinking, what is the importance of the size of the neighborhood you took for your classification? Yeah, okay, okay. Oh, sorry.
Hello, thank you for the great talk. On the topic of the glioblastoma, that I believe that there are also like methylation classifiers that work quite well in classifying this data, and I believe you now mainly presented an RNA-seq based classifier. Would it be possible to maybe also integrate this like data layer into it in terms of like improving maybe also some of the omics layers you get in?
Yeah, exactly. I think I've seen some wonderful poster at this conference, and we're going to collaborate. In fact, the data science hackathon the students were involved in, they did more than the transcriptomic, but I'm limited by time, so I didn't show other markers. In fact, if you link other markers, you also find additional validation. We actually found a virus link to one of the genes in the markers, and we're hypothesizing that this gene's expression can either protect from the virus infection, infections, and therefore making the patient more or less sensitive to the disease progression, onset. There are many, many interesting things that we can hypothesize and build. Yeah, you make a great point. Thank you.
Thank you.
Hi, thank you. Very nice talk. So TSNE, especially in relation to UMAP, doesn't really retain global structure that well, right? So do you have any specific way that you, like between seeds or different runs, do you like track samples, like making sure that, you know, the sample of interest that you were showing really is around the same samples as before? Like, how do you do that?
Yeah, in fact, yeah, that's good. In fact, we allow any kind of embedding in seeds. So you can use a TSNE, UMAP, anything that doesn't matter. We also provide, in seeds, we provide the embedding of 3 different methods. So that, no embedding is perfect. So they're all based on some domain agnostic assumptions. That's why we have to refine it by, once we discover some interesting information, we try to refine it, generate new embedding.
So you incorporate that in your tool, Cs?
Right.
Okay.
Not in the refinement is not incorporated yet.
Okay.
But we probably should do that.
Thank you.
Okay, so we will move to the last speaker. So thank you again, Jake.
Thank you.
And our last speaker is on electronic health record. It's Jacqueline Beinecke. Thank you.
Yes, hello. Thank you very much for the introduction and giving me the chance to present my work here. This is a work I did together with the University Clinic Marburg-Gießen, who provided us with the data and expert knowledge on how to evaluate. I will first go through like a motivation why we even did this work, and then the data, our prediction workflow, results, comparison, and also the limitations of our study. So for the motivation, prostate cancer is the second most commonly diagnosed cancer in men worldwide. The treatment options are radical prostatectomy or radiation therapy, so very severe treatments, and still with these treatments there is a high chance for recurrence, and the main indicator for this is an increase in PSA serum concentration, which is a prostate-specific antigen. And the problem is when so in the early stages of recurrence, the PSA values are quite low, and so it's hard to detect the recurrence at this stage, but it's very critical to find the recurrence very early on, and that's why the PET-CTs in this case are used as an additional diagnostical tool. And so we are focusing on predicting the outcome of the PET-CT, so whether the PET-CT will show a positive recurrence or not. This is mostly motivated by the fact that PET-CTs are very expensive, so it would be great if we could just do a test and predict if the PET-CT would be positive or not. And so, yeah, for the data, our data is from the University Clinic Gießen-Marburg, and it's electronic health records. We had to do a lot of cleaning, and at the end we had 240 patients and 22 features, which I know is not a lot, but we also had some trouble with the data, like high imbalance. So we had 208 recurring patients and 32 non-recurring patients. This is mostly because the PET-CTs are done when the recurrence is highly likely. And we also had to deal with a lot of missing values. So in the whole data set, we only had 11 patients with complete information. So to combat these 11 patients with only complete information, we looked at the features, and we decided to remove all features that had less than 20% missing values. So in the end, we remained with 10 features, and now we had 146 patients with complete information. And the features included things like the PSA values, the doubling time of PSA values, also Gleason score, and some more. Yeah, and to our prediction workflow, so at first we split the data into training and test by 80-20, and then we looked at some pre-processing techniques because, as I said, we have very highly unbalanced data and also a lot of missing values still. So we mainly focused on upsampling, which increases the smaller class by a certain percentage by looking at the k-nearest neighbors of each sample. So we used this to upsample our smaller class by 20% using SMOTE. And then we also did imputation with random forests to remove our missing values, and we wanted to see if this helps. So this method is random forest impute, and it first imputes the missing values with medians and then uses the proximity matrix of a random forest to impute the medians. Yeah. And after the pre-processing, we trained different models. So we looked at logistic regression, random forest, gradient boosting, and support vector machines to have a variety of linear, nonlinear models to evaluate. And then we used a lot of metrics, mainly sensitivity, specificity, to see how good the models are at classifying positive and non-positive patients, and AUC to see how good the models are at distinguishing the two classes, and the area under the precision recall curve and the Matthews correlation coefficient as metrics that are better with unbalanced data, which we had. And in the end, we repeated this workflow 100 times, which is called Monte Carlo cross-validation, which has a lower variance in the end, so we can compare the models better. And to our results, so with no pre-processing, most models performed equally well, so around a little higher than 75% AUC. Just the support vector machine had a much higher variance and also lower mean of AUC. With upsampling, looked similar. Nothing much was changing. With imputation also, though here some of the models were a bit on more equal ground. And with the combination of upsampling and imputation, also not much changed. So in the end, the upsampling itself improved no model significantly, so p-value larger than 0.01. The imputation only improved the sensitivity significantly for the support vector machine and logistic regression, and imputation and upsampling combined improved the AUC and AUC under the precision recall curve only for the support vector machine significantly. So the takeaway is, for our data, it didn't do much, it just improved the support vector machine, which was the worst performing model. And yeah, so overall results, AUC, our highest mean AUC was 78% with the random forest, and sensitivity and AUC under the precision recall curve was very high, mainly because we have a lot of recurring patients, so a lot of positives. And as expected, specificity and Matthews correlation coefficients were quite low, so our models all had problems identifying non-recurring patients, which is probably due to the fact that we did not have a lot of non-recurring patients. And to put it in comparison with other work, so Zagos et al. Trained a convolutional neural network on EHR and postoperative data, and they predicted, like a lot of other people, biochemical recurrence, which is the increase in PSA values, and they achieved an AUC of 0.77, which is a little bit lower than ours. And Li et al. Trained gradient boosting algorithms to predict the recurrence after 3 years and 5 years, also on electronic health records, and they achieved AUC scores of 80 and higher. And Kumar et al. Also did imaging confirmed recurrence, but on H&E stained tissue, and also used a convolutional neural network, and they achieved an AUC of 81. So we are right in there with our performance. And I have to say, most of these had larger data sets. So the Li et al., they had from the Korean database over 1,000 samples, and Zagos et al., I think, also had about 5,000 patients. So their databases were a lot larger than ours. Yeah, so to the limitations of our study, we still need larger data sets to confirm our findings. And our results that specificity and Matthews correlation coefficient values are quite low. We think it's because we don't have a lot of non-recurring patients, but this also, we need a more balanced data set to see if that's really the reason or if there's a problem in the information in the data for this. And then the PET-CT itself has lower soft tissue contrast and resolution than, for example, MRI data. So there's also the possibility that maybe one could evaluate the recurrence using different kind of PET-CTs or MRIs in that case. And yeah, for my acknowledgements, I want to thank the University Clinic Gießen-Marburg. They provided us with the data. Patrick also did his PhD in medicine, so he had all the hard work of calling all the doctors and getting all the information. And Professor Haider from the University Marburg, who was my former PI and who had an overviewing position, and then my current PI, Professor Anne-Christine Hausfeld from the UMG, who was like the main manager of the whole project and kept everything together. And yeah, while I have your attention, a short announcement. We have currently in our lab in Göttingen, we have open PhD and postdoc positions in the field of federated learning. So if you are interested or you know someone who's interested, feel free to contact me or my PI. And yeah, thank you for your attention. And yeah.
Thank you. Questions?
Thanks for your talk. If I understand correctly, the best performing model, let's say, in the previous work, is that for Korean? Is that Lee et al., etc.? What kind of data did they use? Is it clinical data only?
It was a Korean prostate cancer database. So I think they were only Korean patients.
So what's only clinical data, right?
Yeah, electronic health records.
So how do you explain that? So what features did they find, the clinical features that are predictive, which actually performs better? Of course, they have better performance because they have a bigger data set, but you can also learn, or we can learn from that study. So those are not done for nothing, just for that publication. So what did they learn? Can't you, or didn't you try to use these features, which are, I hope, available in your data set to predict, then you can compare whether these in the PET-CT features are actually useful?
Yeah, so they had more features in their database, and the problem is, for us, was that some of these features, for us, had a lot of missing values. So we just didn't have enough, sadly, for those. But I think they had a more, also like, what did they use? They had tumor size and some information that, for us, sadly, a lot of missing values. But yeah, you can, of course, read their paper. They also found a lot of interesting features.
Okay, so if I take home message, so for the patients, actually, so scientists can learn from those bigger studies with the clinical features, which are, I think, easier to obtain. But maybe you explored the features from PET-CT scans, which can be added value. So it's just how I see it, the result of your work. Well, thank you.
Thank you.
Yeah, one question could be like, if you can have more patients, more features, or less missing data, what would be your choice in between the three?
I think less missing values and
So first less missing values.
Yeah, then more patients.
And then more features.
Yeah.
Okay. Yeah, that's good. Other questions? Okay, so if not, we'll thank you, Jacqueline, again, and we can go for coffee break.